Welcome to our seminary. Global Genome Profiling reveals DYRK1A as critical activator of the human mitochondrial informationary, an article from the magazine Nature Communications. We are Mariana Araiza and María Isabel Arango from Universidad Pontificia Bolivariana, medical students of third semester.
For beginning, we are going to start talking a little bit about mitochondria. This one is found in the cytoplasms of almost all eukaryotic cells. They are oval in shape and they are composed of two distinct membranes. The outer mitochondrial membrane is freely permeable to small molecules and contains special channels. In contrast, the inner membrane is far less permeable, allowing only very small molecules to cross into the matrix. The primary function of the organelle is generate large quantities of energy in the form of ATP. This process, known as chemiosmotic coupling of oxidative phosphorylation, powers nearly all cellular activities, including those that generate muscle movements and brain functions. Also, mitochondria store calcium for the cell's signaling activities, generates heat, and mediates cells growth and death.
In mitochondria, we found the import receptor TOM70, which is targeted by the kinase DYRK1A. This modification plays a critical role in the activation of the carrier import pathway. Phosphorylation is strongly required to stimulate the transfer of precursors from the receptor-bound state to the translocation pore. Impairment of DYRK1A signaling leads to compromised mitochondrial structure and function.
Final objective. Proof global genome profiling reveals DYRK1A as critical activator of the human mitochondrial import machinery.
Let's take a look at the methods that were used in the study. The first one is QRT-PCR or real-time PCR, which is a laboratory technique of molecular biology based on the polymerase chain reaction. It is a variant of the conventional PCR. It is quantitative. It allows to quantify in addition to detecting and amplifying DNA or RNA sequences in real time. That allows us to measure the amount of DNA fragments that are produced in real time and not only as it ends, as in conventional PCR. Electrophoresis is a common lab technique used to identify, quantify, and purify nucleic acid fragments. Samples are loaded into wells of an agarose or acrylamide gel and subjected to an electric field, causing the negatively charged nucleic acids to move toward the positive electrode. Shorter DNA fragments will travel more rapidly, whereas the longest fragments will remain closest to the origin of the gel, resulting in separation based on size.
Immunofluorescence. It is based on the union between antigen and antibody. In this one, the AC binds to a molecule of fluorochrome that absorbs light at 200-400 nanometers and emits wavelength greater than 400-700 nanometers. Most used fluorochromes are fluorescent isothiocyanate, rhodamine, and the immunofluorescence it could be direct and indirect. In the trial was used to isolate the TOM complex. Immunoprecipitation. It is based on the formation of insoluble complexes, the reaction of antigens with multiple antigenic determinants against antibodies causes precipitation reactions. This process can be used to isolate and concentrate a particular protein from a sample containing many thousands of different proteins. In this article was used to analyze mitochondrial morphology when DYRK1A is inhibited.
To study the activity of the kinase, we incubated TOM70CD in the presence or absence of DYRK1A. I monitor phosphorylation activity via phosphate affinity. TOM70CD migrated slower upon incubation with DYRK1A, indicating a dependent phosphorylation of TOM70CD. In contrast, the receptor domains of TOM22 and TOM20 did not show a change migration upon incubation with DYRK1A, indicated that exclusively target the TOM70 receptor. In the electrophoresis of non-treated mitochondria revealed a prominent TOM70 immunoreactive band that was accessible to treatment with lambda phosphatase, which we can see in figure 1D. This indicates that TOM70 is almost quantitatively phosphorylated in vivo. Then we analyze if indie mediator inhibition of DYRK1A would indeed cause an impairment of TOM70 dependent protein biogenesis. It was tested by importing radio-labeled TIM23 precursor, a model substrate of the TOM70 dependent carrier import pathway, into mitochondria that were isolated from cells grown in the presence or absence of indie. Surprisingly, import of TIM23 was only mildly affected.
In this graphic, the scientists analyzed if the inhibition of the gene DYRK18 will really cause a deterioration in the biogenesis of TOM70. They used indie as an inhibitor. So, in the image below, they were analyzing TIM23, that is a precursor of the protein TOM70. They treated with the inhibitor indie, but the concentration of TIM23 didn't change that much. In the image D, the left first image, it shows us the phosphorylation portion of TOM70. The second line looks darker, and that means that the phosphorylation portion in TOM70 decreased with the inhibition of indie, but the image at right show us that the concentration of the protein TOM70 actually increased with the inhibition. In this graphic, they were analyzing the import way using TIM23 to do the import and the specific point of phosphorylation in TOM70. And the results are that the phosphorylation in the point SER91 ALA is the most important one. That's why in both of the images we can see these lines darker. That means that the concentration there is higher than in the other phosphorylation points. That's why they conclude that even if the rest of the phosphorylation points in TOM70 aren't phosphorylated, SER91 ALA is capable of maintaining the import weight by themselves.
For the discussion, we have three authors. Let's start with Young, who proposed that ATP hydrolysis is required for the release of chaperones from the precursor. However, in our study, we offer an exciting alternative explanation that the ATP dependency is mediated by the protein kinase DYRK1A discovered in this study. Then we have Edwards, who says that only a very limited fraction of the human genome is investigated in detail. This is true. The human genome comprises 550 different kinases. The DYRK1A is a beautiful example of the importance of these kinases in the discovery of signaling pathways that regulate mitochondrial protein biogenesis. Finally, we have SOPA. DYRK1A is involved in any different cellular processes, including cell differentiation, proliferation, and survival. And it is true, DYRK1A is a critical activator of the human important machinery, and its dysfunction is described to cause an array of disease phenotypes, including intellectual disability, speech delay, motor deficits, and autism.
Conclusions. Molecular biology is a fundamental area of medical knowledge because it provides us with the necessary tools to understand the process that occurs in the human body, from the function and interaction between the molecules and cells that compose it, which allow us to understand exactly where the damage is found in a pathology and therefore develop better mechanisms to fight them. The process developed from the molecular biology have allowed the study and manipulation of DNA that has opened the door to gene therapy, which will allow to definitely cure multiple pathologies. This is currently one of the areas of greatest interest in medicine, and in turn one of the most promising. Regarding the article previously exposed, molecular biology allows the identification of DYRK1A as TOM kinases, than the relation between this gene and the correct function of the mitochondria, with the appearance of phenotypes such as the spectrum and Down syndrome.